So my name is Christy Jones. I'm the Medical Director at Muscular Dystrophy New South Wales and work closely with the team to put the seminar together. So before we start, we'd like to acknowledge the Aboriginal and Torres Strait Islander people as the traditional custodians of this land. We acknowledge the Wanagal of the Eora Nation as the traditional custodians of this place that we call Sydney Olympic Park. So I will move straight on and welcome our first speaker, Sam Bryan, who is our, I think, fifth PhD student. And we received the Sue Connor bequest some years ago, which was an incredibly generous bequest that allowed us to support several bright young stars to start off in their PhDs. And we've been lucky enough that many of them have, having had a start from our bequest, gone on to get NH and MRC funding, which is very highly competitive funding, which has of course allowed us to support more PhD students. So it's been a wonderful program. So Sam is out in her first year of her PhD at the Children's Hospital at Westmead. Come on down.
Thanks, Christy. Today I'm just going to be talking about what our group and what my PhD is focused on, and that's splice variants and trying to make sense of these variants. To outline what our group does is we have a research cohort of neuromuscular disorders, patients with neuromuscular disorders, and we're looking through the genetics of these patients to find out what the answer is and why they have this neuromuscular disorder. So the importance of getting this diagnosis is that it guides clinical management of patients, it can pinpoint certain treatment methods, and it can allow for the eligibility of certain clinical trials that might be available, and allows for family planning. Finding this genetic diagnosis, however, can be extremely difficult, as each person has thousands of unique variants that might be unique to them and their family. So trying to sift through them versus what's occurring, what variation is occurring naturally can be really difficult and challenging to understand what the answer is. So we have a research cohort of roughly 200 families, and we've only found an answer for 60% of these families. Of this 60%, we've found that 1/3 of patients will have at least one splice variant. So my PhD focuses on these splice variants because they're extremely difficult to predict whether or not they are the disease-causing variant. It's really difficult to understand what sort of impact one of these sort of variants is going to have. So let me explain to you what splice variants are. So to start from the very beginning, DNA is like code. In the same way this code is coded by zeros and ones, DNA is made up of A, G, C's, and T's. This exists in a linear format in your cell, and it binds to a complete copy, a complement of that exact sequence. So we see that every C and G pairs together, A and T's pair together, and you end up with this ladder-like formation that winds itself into this double helix that some of you might be familiar with seeing. You actually have 3 billion of these base pairs, these A, G, C's, and T's that make up you. These are arranged into chromosomes. So we see we've got 22 pairs of chromosomes and then our sex chromosomes. And you've inherited one chromosome from your mom and one chromosome from your dad. And amongst these 3 billion base pairs of information are your genes. So your genes encode for proteins, and those proteins make up who you are. There are 20,000 genes that are known within the human genome scattered throughout these 3 billion base pairs, with 3,200 of these implicated in genetic disorders. For neuromuscular disorders, we found over 600 genes so far that are associated with neuromuscular disorders, and we continue to discover more. Here on the screen, you can see that I've highlighted some of the genes we've been interested in over the years and where exactly they are throughout the genome. So you can see just how scattered they are through these 3 billion base pairs and how hard it can be to look through all of these different areas to try and find the answer for each individual. So today I'm going to be focusing on 2 genes that I've been working on recently. They're on the X chromosome here. So to explain splice variants, I'm going to refer to these 2 genes for context. So here is the EMD gene. So EMD codes for the protein emerin, and emerin is an important a protein in muscle that helps for good, strong muscles. it has 2,200 base pairs, and these base pairs are the AGCT code I was referring to before. So this big blue box represents that code. But what's actually needed for the Emrin protein itself is only 765 base pairs of this code. So these introns are this non-coding region that actually aren't needed to make the protein Emrin, but yet they're still in there amongst the code that we do need, these exons, that do contain the important information to make the protein Emrin. So by comparison, DMD, a gene implicated in Duchenne's muscular dystrophy, which some of you may have heard of, it's, by comparison, is a massive gene. It's 2 million base pairs long. But when we look at what actually is needed for the protein dystrophin, we only need 11,000 of those base pairs to actually make the protein. We somehow have to sort through this 2 million base pairs of information and take away the non-coding information that we don't really need for the Emrin protein. So the process by putting those blocks together, those blocks of useful information, to glue them together is the process of splicing, which is what I'll be referring to a lot. So interestingly, only 2% of the 3 billion base pairs I was referring to before actually encode for proteins. So how this process works is you have your DNA, your 3 billion base pairs, and to actually make a protein in the end, you want to, your cell makes a copy of the DNA, and then it edits that copy to suit it for whatever job it's going to do, for what protein it's going to make. So an analogy for this, if you imagine like DNA as a blueprint for a house, for instance, and a plumber's come on site and he wants to just work on the plumbing in the bathroom. He's got this massive amount of information in this blueprint for the house that he doesn't really need. He doesn't really care about anything else that's happening in this house. He's only interested in the plumbing in the bathroom. So he's looking at these instructions, he's like, nah, I don't really need all of this. He's going to cut it up, he's going to edit it, he's going to take just the instructions that he wants, the instructions he's interested in. And now he's got just a nice little condensed version of the original instructions, and now we can go to the building site and build the plumbing. So it's similar to how our cell looks at our DNA, takes the information it wants, leaves the rest, and goes on to make the protein. So when I was talking about those blocks of information that need to be glued together, so these exons, these coding bits of information that actually make the protein, need to come together, and this intron, the non-coding information, needs to be cut out. So what happens is there are important sequences within this non-coding region. You can see the GT and the AG there. There are these protein complexes in your cell that come and bind to these particular spots. So each protein will have an affinity with some of the code that's in the non-coding region, and it brings these two exons together, and this non-coding bit of information that we don't want because it's not useful to us is excised, and you're left with the two blocks of information that we care about stuck together in a cohesive fashion so that the cell can then make the protein that we're interested in. So mutations in this intron here, see there, so anywhere within this non-coding region can affect how these proteins actually bind to that intron. And so when we see a mutation in a patient within this region here, it's a question of, will it affect how these proteins bind or will it not? And that can be the really tricky part of what we do, and can be very confusing for doctors and for patients who are trying to interpret this genetic information and decide whether or not we think this might be the cause. So here's an example of how devastating splice variants can be. So if we go back to our plumbing example before, here's our gene that has our plumbing instructions. When we correctly splice all these blocks of information together, we see that it reads insert pipe A into pipe B, then secure using screw C, stop. So the plumber has this bit of information, he can go into the bathroom and he does those things, we have a good outcome, the bathroom works. However, if we were to lose this block of information, this coding block here, the new sentence might read something like, insert pipe A into P head, secure using screw C, stop. See, that makes a little bit less sense. Maybe the plumber might be able to go into the job, maybe try and fumble around, maybe get some sort of outcome, but we're not really sure. Like, it's going to be hard to determine whether or not the plumber's going to do a good job. What if instead, when we were editing the instructions here, we included some of the nonsensical information that we didn't want to include, that stuff that's supposed to be excluded, if we accidentally include that instead, then we might read something like this. We instead get this nonsensical nonsense mumble jumble followed by a stop. And that stop means that the plumber no longer needs to finish reading the rest of the instructions. These instructions are gone, and that's it. We've got an early stop to our thing. So at this point, the plumbing job might not go so well after all. So for each splice variant that happens, every splicing mutation that occurs, 2 out of 3 times we'll end up with this premature stop that prevents the rest of the instructions from going to the cell to actually make the protein that we're interested in. So how do we actually determine whether a mutation within that non-coding region will disrupt this process, this important process? And we need to be actually looking at this messenger RNA here. A lot of genetic testing that's done will look at the DNA first, but then if you don't have any answers, it's helpful to look at this messenger RNA so we can actually see what instructions have been edited how and how that might implicate disease. The problem is to get messenger RNA that's relevant, we need to actually get the RNA from the tissue of relevance. So in this case, it would be muscle. So you need a muscle biopsy to be able to take that RNA. And then that RNA itself is quite unstable and needs to be carefully handled, so that can make transportation difficult. But nonetheless, we can actually study this messenger RNA and see what instructions are being used. So here's an example of a case that we've diagnosed in the past month. It was a Duchenne muscular dystrophy case. So as some of you may know, Duchenne muscular dystrophy is the most common muscular dystrophy. There's 1 in 5,000 males being affected. And here's the gene I mentioned earlier up here, DMD, which is 2 million base pairs long. So it's a lot of information to have to trawl through to try and find the answer. DMD presents at 2 to 5 years of age. There's delay in gross motor mile skills, limb girdle muscle pattern of weakness, absence of dystrophin in muscle, and that really is a good indicator that we should be looking in the DMD gene, and an elevated serum creatinine kinase. So in our case, this was an adult male who was diagnosed with Duchenne's at around 10 years. So this is an example of how long it can take to get a genetic diagnosis in some cases. So it's a good 20 years before we were able to find this. So there's absolutely almost no dystrophin could be seen in his muscle. DMD was sequenced multiple times and nothing was found. So this was done by several different labs and we just couldn't find anything at all that was going wrong. MLPA was used, which is a technique that looks at big deletions or duplications, really obvious big structural things, but that was negative as well. Couldn't find anything on that. Then we tried the Perth panel of neuromuscular disease genes, which would also look at dystrophin again, but also a whole host of other genes that might implicate some sort of answer, but nothing again. So we looked at the RNA instead of the DNA. So I had the muscle biopsy and I extracted the RNA and we sent it over to our collaborators at Perth and they sequenced the RNA. And what I found in this region here, so I've just highlighted the region that as I was going through the sequencing files, I found something interesting. So here we've got, expanding this section here, we see that we've got exon 23, 24, and 25. See there, we're now running in the opposite direction, I apologize, but these blue boxes represent that coding bit of information that is needed to make the dystrophin protein. So they're the blocks of information we're trying to include. A healthy control will look like this. You can see that we've got this peak height underneath where the exon sits. So this peak here represents that we've got a lot of evidence to say that exon 23 is there. These loops, where you can see 302, there are 302 evidence to suggest that it's going from exon 23 to 24, and so on. You can see how it keeps looping to each exon, and that gives you evidence that those exons are coming together correctly so that you've got the correct amount, the correct instructions to make dystrophin. When you look at our patient in these files, we see that, yes, they go correctly from exon 23 to 24, then from 24 to 25, 25 to 26. But when we get to here, instead of continuing on to 27, most of the time we'll pick up a random bit of non-coding information. So we're in an intron here that, this non-bit of coding information should have been completely ignored, but for whatever reason, in this case, it's being picked up and included in this transcript. So this is fake code that does not belong in our actual instruction. It's going to confuse the plumber downstream. So the next question is, why is this doing this? How come we have this bit of information being used? What's going on here? And so we're able just to sequence this region here. So now that we know where we're looking in the RNA, we go back to the DNA and we zoom up on that specific region and sequence just that specific region to see what we find, see if there's any sort of reason why this might be happening. The only thing we found here, if you see, this is the non-coding information, and this is what the code looks like here in the non-coding. And the blue is where we start including accidental code, that fake exon I was referring to before. The only change we found was this G here. You can see this G I've put in bold. That G in our patient became a T. So that is such a subtle change. It is one SNP, and it's even not anywhere near where our fake exon begins, which is a bit odd. And for someone who didn't have this RNA and wasn't looking at the already edited instructions, if you found this change in the DNA, you wouldn't know what to do with it. You'd have absolutely no idea. So when we remember my schematic from earlier, we see that these proteins love to bind to certain code, right? We know that this AG is really important here, and you can see that in the fake exon. There's a lovely AG site that this green one is going to bind to. But the fact that this T is having such a massive influence on the affinity of that binding protein is super hard to predict. There are algorithms out there that try to say whether or not something like that will happen, but it's unless you actually take the RNA and look at it, it can be really difficult to tell the family something useful from that information. So what does this mean for the dystrophin made by this patient? So all I've told you is that we've got an extra little bit of information, but this somehow has this massive effect. So let's have a look at that. So back to our DMD schematic. So see all the coding, the blocks of information that code for our protein. When those are put together, it will look something like this, a tiny little small amount, only 11,000 base pairs at this point. So splicing occurs. But in our patient, we've got this fake exon, and this fake exon has the code that says stop. So just like the plumber, it's going to get to this point and be like, oh, no, I don't need the rest of the instructions now. I don't need the rest of this protein, so we'll get rid of that instructions. But then the cell notices that this stop position is in the wrong place. We need to destroy that bit of instructions because it's incorrect. So that gets destroyed as well. And then we're left with nothing. So when we look at the patient's dystrophin and muscle, it makes sense as to why you can see in the patient's lane there is no dystrophin, where you can see in the control there's a really big band for dystrophin. So he's unable to make dystrophin because of this one tiny SNP, this one tiny base pair change that led to this massive incorporation of instructions for some reason. So yes, they now have a diagnosis, and that's going to be very good for their family. I just want to quickly go through some other families that I've helped to diagnose using these RNA studies and looking at what the instructions are actually doing. So there were 5 families that we've diagnosed, that I helped diagnose for with Duchenne muscular dystrophy and Becker muscular dystrophy. And because this is an X-linked gene, it's super important for the family because any female in the family could potentially be a carrier. So to give them informed information about their genome can really, really help for family planning purposes. Another 2 families, one with a DOX7 congenital myasthenic syndrome and another with Emery-Dreifuss muscular dystrophy. So an interesting thing about these mutations is that this led to a new mechanism for how we understand splicing works. So they sort of led to a better understanding of really important factors of that non-coding information and how that can lead to problems. So that's been really great, and that will help a lot of families down the track. Particularly now that the family with congenital myasthenic syndrome knows that the gene behind the condition was DOX7, they've been able to be treated with salbutamol. And they've gone from being completely wheelchair dependent to being able to walk unassisted for short distances, which is fantastic for them. Yep, there was those ones. And then Titin is another gene that I really like, the TTN. it's an absolutely massive gene, and it's super complicated because depending on what cell you're looking at and what tissue you're looking at depends on what codes of information it's going to use. So it's completely unpredictable and really difficult to work with, which is challenging, but rewarding when you can find answers. So this particular family had arthrogryposis and myopathy, and we found this variant in a region that we didn't think was particularly important, and we showed that yes, the RNA is using that region, using that region, but it's not using it correctly. It's messing it about. And then we found that this was actually a fairly common variant, and we found 7 other families that had this variant, and that led to the diagnosis of all those families because of this aberrant thing. And it's just great that we're sort of understanding now that that region of that protein is actually important, and this might help diagnose even more families later on as people know to start looking at this region rather than other regions. And then there's a few other cases that I've listed there where I've followed up RNA studies on variants that clinicians and doctors have found, and able to show that yes, that indeed is the problem. So yeah, in total so far I've diagnosed 18 families and 23 individuals so far. So the goals of my PhD, going back to what I'm doing here, is to improve our understanding of what the code in these non-coding regions actually does for the splicing of these exons together. So we're studying RNA from patients who have intronic mutations to get a better understanding of like what code is important and needed to actually get rid of the non-coding information correctly. We're also going to be using cell-based assays so we can edit the code as much as we like and just see what happens, and then that'll give us better understandings of what's happening so that we can inform new algorithms to better predict what's happening. And our lab is currently working on an algorithm that's competing other algorithms for just one side of the just at one point in the intron. So it's really promising work that we're doing, and the more studies that we'll be able to see in patients and see what's happening will lead to more information into that algorithm, which means that we can further predict, and maybe in the future muscle biopsies will be less necessary, hopefully. So the overreaching goal of our group and my PhD is to provide doctors with tools and services to be able to better understand what's going on so they can get back to the patient and say, with informed information, I think this is the answer for your muscular dystrophy. So that's the overall goal. So I just want to say thank you, Muscular Dystrophy New South Wales, for providing me with the scholarship to allow me to do these studies. Yeah, I really appreciate it, and it's helped out a lot. And I want to thank everybody in my lab who've helped me with this PhD, as well as our collaborators in Perth and overseas who are working. We're all working together to try and figure out what this non-coding bit of information is important for. Thank you.
Thank you, Sam, for making such a complicated topic so very, I think, accessible. Has anyone got any questions for Sam? Yep.
Is there any chance that you'd be doing other types of muscular dystrophy in other genes? Or?
Yes. Whenever we have a patient and we're looking at their genetic information, we generally try to look at all of the genes or hone down on what genes we think are likely. But yes, at the moment we sort of find a splicing variant, and it doesn't really matter what gene it is, so long as it might be. So yes, we're looking far wide.
I think that's one of the wonderful things about this. It's applicable to every gene.
Yeah.
This concept to every gene. So
and every genetic disease as well. So we're actually starting to pick up cases from other types of genetic disorders that affect the brain or the kidney or liver. Like we're really starting to move out as well. But muscular dystrophies is easier for us to study first because a lot of patients have already had a muscle biopsy, and we can look at those instructions in that muscle biopsy. So we're a bit ahead in the muscular dystrophy field because we actually can look at the instructions easier rather than getting brain tissue or kidney tissue.
Well, I was just interested in gene 14. I noticed you didn't have those arrows going to gene 14.
Oh, the curve, yeah.
That's not one of the ones you've done so far. So what that means?
No, that's just me being, I just picked like 20 of my favourite, and they just mapped to those areas. Yeah.
She'd be fun on a Saturday night, wouldn't she?
Yeah, so there's actually 600 genes or 600 more genes that I didn't actually bother with the arrows, and they're spread out through that. So yeah, there's quite a bit of knowledge, but it's still hard to trawl through.
Thank you.
I'm just very curious, how did you arrive at salbutamol as a correct, appropriate treatment for the young children and wonderful result from it? How did you arrive at salbutamol?
So I didn't personally arrive at that. They were previous studies done, and I think it was to do with an asthma case.
Christina might be able to answer that. Do you know how salbutamol became the treatment for DOX7? Was it?
I don't know how it started, but basically if it's DOX7, if you use Ventolin, it's quite cheap. So most clinicians, even if without the genetic diagnosis, is happy to try it. Side effects is low, and it does work very well with DOX7 CoQ as well.
I think somebody noticed that a person being treated for asthma improved their muscle weakness.
That's unbelievable.
Yeah.
Thank you.
Can you please comment on gene editing in this context and techniques like CRISPR-Cas9?
Yeah, so if you do have a genetic diagnosis, I think it could be possible in the future for CRISPR-Cas9 to sort of implement so that you could go into where the problem is and edit it and put in correct sequence. So that's something that's possible. Something that's happening sooner though are morpholino treatments where basically you have a bit of sequence, that synthetic bit of sequence that you've made that can go in and block the splicing of one thing. So for instance with the patient that I showed you before, and they had a that bit of information that shouldn't be been included. What could potentially be viable for him in the future is that you have a synthetic bit of DNA that binds to that and prevents it from being used, so that therefore thereafter any transcript that's made, any messenger RNA made from those instructions won't happen. So that's something that we're looking into, but it's a bit
And I
far
think
away.
these type of changes are very amenable to those type of treatments. It's a little way off, but it's exciting.
Yeah, and it will be very patient specific. These treatments will have to be very tailored to the exact mutation that they have. Yeah.
I think we might move on. Thank you so much, Sam. It was fascinating.